Today we are going to study about the few more parasites and their life histories which are also responsible for causing diseases in man. Let's move on to the today's some of the life histories of the parasites. Ascaris is a lumbricoides. It's one of the common parasite which is commonly called common round worm, which belongs to the phylum Nematoda and the class Phasmidian. Ascaris lumbricoides, commonly called a common round worm, and it is one of the most common enteric parasite of man, which enteric means it's which mainly resides inside the small intestine of a man, and it's a monogenetic parasite which completes its life cycle in single host, and it is a dimorphic, so that we can able to differentiate the easily the male and the female, and that means it exhibits sexual dimorphism. By their external morphological features, it is we can differentiate the male from the female, and it's a pseudo-coelomate parasite. As you know that the phylum Nematoda it's provided with a pseudocoelom inside them. So this parasite belongs to that particular phylum, and it lives inside the small intestine of man, and but especially more frequently it are the more prevalent in case of children. Now it's a cosmopolitan in distribution. It's a worldwide in distribution, present in almost all the parts of the world, and the infection mainly occurs through by means of contaminated food and water. So what are the infective stages of this parasite? And the infective stage of this parasite is the embryonated egg, which is provided with the second stage rhabditiform larva. Embryonated egg with second stage rhabditiform larva. This is a stage which in this parasite enters into the body of host. Now come to the structural aspects of this parasite. The sex, as we discussed earlier, it is a dimorphic. That means the sexes are separate, and it exhibits the sexual dimorphism. A very clear distinct sexual dimorphism is present, which means we can easily distinguish the differences between the male and a female. So some of the common features which are present in both of them are the body of both the parasites is actually the elongated and cylindrical in shape, and in both the mouth is actually present at the anterior end, and at anterior end it is also provided with a 3 chitinous lips. Close to the mouth, very close to the mouth on the midventral side, they are provided with the excretory pore. These are some of the being cylindrical, long, elongated, and pointed at the both ends, and mouth is present at the anterior and provided with the 3 chitinous lips, and also midventral on the anterior side provided with the excretory pore. These characters are common to both male as well as female. Let's move on to the some of the differences that are present in between the male and female. How do you can able to differentiate the male from the female? So if you look at the male, at the posterior end of the male, which is considered as a tail, actually it's not tail but considered as a tail, it's a posterior curved tail is present, and also provided with a female, also provided with present on the midventral 1/3 of the mouth, and the anus is a little bit present at the end, and it has got the straight tail, it has got the curved tail is present, that is by which we can able to differentiate straight tail and the curved tail is present. And whereas for the female it's provided with the anus is present over here, but on the almost at the 1/3 length of that for the female provided with the female genital aperture is present. When it comes to the male, it is actually provided with the cloaca is present, and in case of the Ascaris. So that is the difference. Cloaca here in male and the anus is in the female, and the female genital aperture is almost present at the ventral side of almost at the anterior 1/3 from the anterior end. So like this we can able to differentiate between the male and female. Now move on to the life history aspects, and now both resides within the small intestine of man, where the act of the copulation occurs. And now after the copulation, the female is responsible for the nearly releasing of 2 lakhs eggs per day. Now the eggs are actually get surrounded by externally by, and the egg is actually surrounded by a protein coat, and below that one you will find another layer which is the chitinous shell in between the middle and towards the inner side you have got the lipid layer is present. Like that, there are the 3 layers are present. So being the outer egg, it is a protein layer which is wavy in margin. These eggs are called mammillated eggs. The eggs are named as mammillated eggs. So inside that one, anyway, the nucleus and the cytoplasm is present. So protein layer and the lipid layer, chitinous layer and the lipid layer, these 3 layers are present inside that one. So entire that outer protein layer being with the rippled surface, and so we call them as mammillated eggs. Now these eggs and such type of the eggs that are get liberated into the lumen of the intestine, and they comes out along with the fecal matter, and these eggs comes out along with fecal matter. Now the development actually started when they when they are present within the eggs only. So now as the development starts, it results in the formation of the first stage rhabditiform, the larva. The inside the eggs, the first stage that is formed is called rhabditiform larva. Now this rhabditiform larva, which undergoes the first molting. So molting is shedding of its outer exoskeleton, what we call, to facilitate the growth of these stages or the larva. So outer exoskeleton is shed off and to facilitate the growth. That's what we call it as molting. So after this first stage larva or the first stage rhabditiform larva undergoing the first molting, it gets transforms into the second stage larva. So this second stage rhabditiform larva present inside the eggs, which is actually regarded as infective stage to the man. Now as they comes out and they reaches the new host, that is a new man, through the contaminated food and water. Now whenever the these embryonated eggs and almost and ascaris, and how long they can able to survive in this stage. And so once the eggs are released outside the body of the host, and after developing into the infective stages, how long in this stage they can able to survive. And so almost they can able to survive for the nearly the 6 years. And so within this period, if they are not transformed into the new host, they will die or perish. Now once they get through the contaminated water and the food, if they reaches to the new body of the host, like especially the man, then it again passes through the different parts of the alimentary canal and reaches to the small intestine. So once it reaches to the small intestine of the man, so after getting reached to the alimentary canal of man through the contaminated food and water, they undergo the extra-intestinal migrations. And so if the conditions are favorable, the embryonated eggs of the ascaris can remain viable almost for the 6 years within the moist soil. Now coming to the extra-intestinal migration, and suppose if you look at the extra-intestinal migration of this parasite, the second stage rhabditiform larva in the small intestine, first it reaches to the hepatic portal vein. Through the hepatic portal vein, it attacks the liver. From the liver, it reaches to it goes to the postcaval vein. This is the vein that connects the liver and heart, and from there it reaches to the heart, and from the heart, as the blood pumps the deoxygenated blood, it reaches to the lungs through pulmonary arteries. From the pulmonary arteries, it goes to the lungs, and from the lungs, where it's inside this alveoli of lungs, and it undergoes the second molting. So after undergoing the second molting, it transforms into third stage larva, and this third stage larva again undergoes third molt. So after undergoing the third molt, this parasite reaches to the fourth stage larva. So from that again, it from the lungs it comes to the bronchi, bronchi to the trachea, trachea to larynx, larynx to glottis. So almost it travels to the entire the respiratory tract in opposite direction. Glottis, and from the glottis it comes to the pharynx. Again from the pharynx it enters into the alimentary canal like stomach, small intestine. After reaching the small intestine, it undergoes the final molt, that is the fourth molting, and develops into the younger stages. And these young ones attain the sexual maturity within a duration of 8 to 10 weeks. So like this, the parasite actually completes its the whole life history. If you just look at into the schematic diagram of this entire the life history of the parasite, see this one, that both the adult tapeworms, the male and the female adult tapeworms, they undergo the copulation inside the intestine of man and releases nearly the 2 lakhs eggs. And so all those eggs shape are being the rippled surface, they are called mammillated eggs. And so these mammillated eggs are comes out along with the fecal matter, and within that moist soil, the development of the parasite gets starts. And so that gets transforms into the first stage rhabditiform larva that undergoes first molting. After undergoing the first molting, they get transforms into second stage rhabditiform larva. So which reaches the new host by means of the contamination. So through the process of the contamination, they reach the new host. Now where inside them the shells get dissolved inside the small intestine, and after resolving the small intestine, they enter into the blood circulation, and from the blood circulation through the hepatic portal vein they reaches the liver. Inside the liver again, from the liver it reaches to the heart through postcaval vein. And from that the heart pumps the blood to the lungs through the pulmonary arteries, so where it undergoes to the lungs again. So from the lungs, inside the lungs of the alveoli, it undergoes the second molting. So first molt actually occurs in the moist soil inside the eggs, second molt occurs inside the fresh host of the man inside alveoli. So which it undergoes transforms into the third stage larva. This third stage larva undergoes third molting, transforms into the fourth stage larva. Totally inside the lungs 2 moltings occurs, in moist soil 1 molting occurs. Now this fourth stage larva now started migrating from alveoli to the bronchi, bronchi to the trachea, from the trachea it goes to larynx, larynx to reaches glottis, glottis to next pharynx, which is a common place for the respiratory and digestive. From the pharynx again travel downwards to the esophagus, stomach, and comes to the small intestine again. And in the so whatever the migration that is parasite actually showing other than the parts of the small intestine, we call it as extra intestinal migration. So now the small intestine, inside this small intestine the parasite undergoes the fourth molting, that is the last molting or the final molting, transforms into the young ones, and again starts the life cycle by undergoing copulations and inside the man. Now like this the parasite keep on completing its life cycle inside the body of the host. And so to attain this young one to the sexual maturity, it requires a duration of 8 to 10 weeks. And now in what way it is responsible for causing a disease, and that is the pathogenicity. And the disease caused by Ascaris lumbricoides is called ascariasis. And if it is asymptomatic if the number of the worms actually exist in less. Sometimes in heavy infections it can cause nutritional deficiencies and also the severe abdominal pain, as they obstruct the path of the digested food inside the small intestine, and small intestine enlarges. So abdominal pain is a common thing, and it's believed that it is actually responsible for the causing of stunted growth, especially in case of the children, as it is believed that it's responsible for the production of certain substances which inhibits the human growth hormone. So they are responsible, it's a belief that secretion of certain types of growth hormone inhibiting factors, so that results in the stunted growth, especially in case of children. Now coming to the prophylactic methods, it is almost same as that of the Entamoeba histolytica that we studied in the previous session, and almost so because it is also responsible for the spreading in the same way, and that is contaminated food and water. So like using the boiled and the filtered water, washing the hands and washing the vegetables and fruits, and also using the septic tanks only for the septic tanks and toilets, and all these are actually the prophylactic methods which are common to both Ascaris as well as Entamoeba histolytica, because both of their mode of transmission is the same, and that is contaminated food and water. So this is a complete life history of Ascaris. So mainly the symptoms of this disease and the infective stages and the disease that is actually caused, number of the molds, where they occur, these are actually the points that are needed to be taken care of. And now let's move on to the another most common parasite responsible for the causing disease in man. The next one is the Uchlaria or Uchlaria bancroftian. So it belongs to the phylum a nematode. It is also another nematode parasite, belongs to the same class, that is Phasmidian. Ascaris Uchlaria, they belong to the same phylum and same class, Phasmidian. Uchlaria bancrofti, it is commonly called filarial vermin and responsible for causing of the disease called filariasis and in human beings, and it is digenetic parasite, like male and female are present. We can easily get distinguish the parasite, and it completes its life cycle in the two hosts, and that's why it is the digenetic. Like female Culex mosquito and man are the hosts required for the completion of the life cycle, and it is also dimorphic. That is, both male and females are separate, and they are very clearly differentiate from each other. And it is also a pseudocoelomate parasite, and pseudocoelom is present inside the body, and it is a histozoic parasite as it lives inside the blood as well as lymph vessels of a man. And so especially it mainly, the adult stages are mainly present inside the lymph of the lymph vessels and the lymph glands and also the lymph. So lymph is being the tissue, it's a fluid connective tissue, so we call it as histozoic parasite. And Sir Patrick Manson actually identified this female Culex mosquito as its secondary as the secondary host, and he discovered its life history inside this particular host. Now come to the structural aspects of this parasite. And sexes are actually get separate, male and female are different, and exhibits the sexual dimorphism is very distinct, and the body is long and narrow and filiform and creamy white in color, and so very long elongated filiform, almost the threads like, and very creamy white in color, and anterior end of the both the parasites are actually the blunt, and the posterior end is being the pointed. These are the common features, and being elongated, filiform and creamy white color, anterior end is blunt, posterior end is pointed. These are all some of the common aspects, and the mouth is present at the anterior end, but without any lips, because in Ascaris just now we studied provided with the 3 chitinous lips are present, but in Uchlaria you do not find this without any lips. In comparison to Ascaris, we say here without any lips. Now if you look know some of the characters of the male form, the posterior end is anyway the curved with a cloaca. It is almost similar to that of Ascaris only, and only in the length and the shapes it shows the differentiation, but the posterior end of Ascaris as well as Uchlaria male is also provided with a curved posterior tail is present with cloaca. A pair of unequal chitinous spicules are the copulatory spicules is present in the cloacal region. A pair of copulatory are unequal, and like this the posterior end is actually curved and provided with a cloaca is present, and an unequal pair of copulatory or chitinous spicules are present, and these are copulatory spicules which are unequal in their length and which are present. Now move on to the female worm. The female worm posterior end is straight. So anus is present near the posterior end, and the female genital aperture is present somewhere at the almost at the 1/3 length of at the anterior and on the ventral side, and almost from the 1/3 length of the mouth on the ventral side of this worm, it is provided with the female genital aperture or vulva. Almost this presence of the anus, straight posterior tail, vulva, they are also present in case of female Ascaris also. But the shapes are somewhat different, and the shape of these worms are actually long, narrow, filiform. They are actually the cylindrical or round, and that is it. But here they are very long and filiform and creamy white in color. That is differences exist in between the Ascaris and Uchlaria bancrofti in comparison with each other. Now the female worms are actually described as the ovoviviparous. The female Uchlaria bancrofti is described as ovoviviparous because they are responsible for the releasing of the eggs in which the life history gets the life history actually gets started, or the development already gets started inside them, and it develops almost into the larva stage. So it is giving neither to the larva nor to the egg, and so that's why it is actually ovoviviparasite. And even though the larva stage is developed inside, the larva do not receive the nourishment from the mother end. So because it is already it's the larva get surrounded by an egg shell, so eggshell is still persists. So it is neither we can consider it as an egg, nor we can consider it as the larva or any other form. So it is an eggshell inside which a larva is developed. This worm is actually regarded as ovoviviparasite, that is female ascaris. Let's move on to the life cycle of this parasite. As we already discussed, it completes its life cycle in the two hosts. One is namely the man and the female Culex mosquito. Now, in man, the male and female worms are found highly coiled together. Like that, they are highly coiled together, and inside the body both are coiled. See this one, long, elongated, and the filiform, thread-like, coiled with each other, present inside the lymph vessels or the lymphatic vessels. Now, the male and female, after they both will undergo the copulation, and the female is responsible for the releasing of sheathed microfilarial larva into the lymph of man. Because it is responsible for releasing of sheathed ensheathed as a microfilarial larva, so it is called as ovoviviparasite. So it is still surrounded all around the body by a loose cuticular sheath, which is nothing but a modified egg shell. So this one started migrating to the blood circulation and resides from the lymph. It goes to the blood circulation, where especially they enter into the deeper blood circulation during the daytime, whereas they started moving to the peripheral blood circulation, especially during the night time between 10 p.m. to the 4 a.m. So this is a time period during which this sheathed microfilarial larva started moving from deeper blood vessels to the peripheral blood vessel. So the tendency of movement of the parasite from the deeper blood vessel to the peripheral blood vessel during the 10 p.m. to the 4 a.m. this phenomenon is called as nocturnal periodicity. So whenever, because this is the time usually the female Anopheles mosquito feeding habits actually get matches. So whenever a female Culex mosquito sucks the blood of such an infected person, they enter into the gut of mosquito. So if not, if the female Anopheles mosquito do not comes and bite such person, so these sheathed microfilarial larva will die within a duration of 70 days. So within a duration of 70 days, if they are not get transformed, the parasite will die within the blood circulation of man. Now after entering inside the mosquito, so suppose if it enters inside the mosquito, so where it goes into the alimentary canal, that is the midgut of the mosquito, where the sheath is get dissolved by the digestive action of the enzymes inside the alimentary canal within a duration of 2 to 6 hours. Now after that, it is now called as egg-sheathed microfilaria larva, and this egg-sheathed microfilaria larva penetrates the gut wall and reaches the hemocoel of a mosquito. So once it penetrates into the gut wall, it enters into the hemocoel, that is the blood of a mosquito. From there it reaches to the thoracic muscles of the mosquito, where it transforms into the next stage called sausage-shaped larva within a duration of 2 days. Now after that, it is called as the first stage larva or the first stage microfilaria larva. Now this first stage microfilaria larva inside this thoracic muscles undergoes 2 molts totally, and within a duration of 10 to 28 days, and transforms into a long slender third stage larva, that is the third infective stage microfilaria larva, after undergoing the 2 molts in a duration of the 10 to 28 days. So this is actually occurs inside this thoracic muscles of thoracic muscles of the mosquito. So, when such mosquito, Culex mosquito, in fact bites a man, this third stage microfilaria larva reaches the or enters the blood circulation, and from that blood circulation it reaches to the lymphatic vessels inside the man, where this inside this lymphatic vessels, this third stage microfilaria larva undergoes the third and the fourth and the last two molds. And first two molds occurs inside the thoracic muscles of the mosquito. The last two, third and fourth molds that occurs inside the lymphatic vessels and the lymph glands, and where they produce get transforms into young filarial worms. And so, once the young filarial worms are formed within a duration of 5 to 18 months, and they attains the sexual maturity, again they freshly start the undergoing copulation and releasing of the eggs. And so, this is what the whole life history, and even like that of the Ascaris, Eukarya is also undergoes the 4 molds. And so, occurrence of the 4 molds is a characteristic feature in almost all the life histories of the nematode organisms, and whether they are parasites or free living, in all of them they actually undergo the 4 molds. And let us move on to the schematic diagram of this one, where the both adult worms are coiled together inside the lymphatic vessels of man, and they undergo the copulation inside the lymph vessels of man, where after the copulation the female is responsible for the releasing of sheathed microfilaria larvae. See this one, entire the larva is actually surrounded by a sheath, and it's a loose cuticular sheath still exists, and that is, which is nothing but considered as an eggshell. So, it still the eggshell is present, and inside them you will find a larva is present, which is the microfilaria larva. So, it's a larva which is not depending on the female, that is on the mother, because still it is surrounded by the eggshell. So, being existence of the eggshell, we call it as oviparous, and still it is attains almost microfilaria larva. So, it is a viviparous. So, together we call it as ovoviviparous. Now, this microfilaria larva exhibits nocturnal periodicity, and so tendency of movement of the larva from the deeper blood vessels to the peripheral blood vessels, especially during the 10 p.m. to 4 a.m. So, during that times, because it matches, the tendency is mainly exhibited by the parasite to suit the feeding habits of female Culex mosquito. So, at such times, if the female mosquito bites and sucks the blood, they enter into the midgut, that is inside the alimentary canal of mosquito. So, after once enters inside the midgut of mosquito, due to the action of the digestive enzymes inside the midgut, whatever the eggshell or the loose cuticular sheath is present, which is get dissolved, and the microfilaria larva is set free. Now, this released microfilaria larva, by dissolving the wall of the alimentary canal, it enters into the hemocoel, which is the sheathed microfilaria larva, now piercing the wall of the gut, enters into the hemocoel. From the hemocoel, it migrates to the thoracic muscles, and after entering inside the thoracic muscles of the mosquito, where it undergoes gets transforms into the sausage shape. So, almost it's a sausage shaped, means it almost look like the inverted brinjal lichen, so which is the first stage larva or the sausage shaped larva. Now, this sausage shaped larva undergoes the first molting, and that get transforms into the second stage larva. Second stage larva undergoes the second molt and get transforms into the long slender infective third stage larva, because it's the infective stage to man. Now, from there it reaches from the thoracic muscles, it reaches to the again the salivary glands, in that the part is called the labium part, reaches the labium. So, whenever such this mosquito bites the fresh human being, again these larvae are released into the blood of the man. So, after they are released into the blood of the man, from the blood circulation these larvae migrate to the lymph of the lymph vessels and lymph glands, where they undergo the third mold to get transforms into the fourth stage larva, and this fourth stage larva undergoes the fourth mold, that is the last mold, and transforms into the young ones. and these young ones attain the sexual maturity in a duration of 5 to 18 months. So totally in this life history also we observe the occurrence of the 4 molts, and 2 in thoracic muscles of the mosquito, another 2 in the lymph vessels of the man. In total 4 molts occurs. Even in the ascaris also totally the 4 molts only occurs, and 1 in case of the mammillated eggs and 2 in case of the alveoli of lungs, and another one occurs inside the small intestine. In total the 4 molts, and so it is a characteristic feature of the all the nematode organisms to undergo 4 molt, whether a free living organism or a parasite. Move on to the pathogenicity of this, how it occurs. And so a light infection causes a filarial fever, and which is actually characterized by headache, a little bit of the mental or the psychological depression will be there, and with the increase or the rise of body temperature is also seen whenever the parasitic infection inside the lymph and the blood are very low. Then this leads to the inflammation, and inflammatory reactions occurs inside the both lymph vessels as well as lymph glands. And inflammation of lymph vessels we call it as lymphangitis, and inflammation of lymph glands we call it as lymphadenitis. And so itis is a suffix for the any inflammatory symptom as it we call. Now in case of the heavy infection, what happens? And so suppose if the more number of the parasites are whose larvae are present, actually larvae are present inside the blood, adult worms are present inside the lymph, and when the both of these numbers are becomes more and more and are get accumulated, the dead worms are also keep on dying inside the lymph vessels due to the obstruction and all. So the lymph and lymph glands started swelling because of the increase in the number of these worms inside the lymph vessels. And so this swelling includes swelling, and so that makes the increases the permeability of, due to the swelling of lymph vessels, increases the permeability of those walls, and whatever the lymph that is present inside started coming out, and this becomes edema, the lymphatic edema, that symptom we call it as. And in this tissue, whatever the tissue one, the fibroblast cells become active, responsible for the production of the more amount of the fibers, so that fibrous tissue also develops inside that area. So and the whole structure and where this severe infection is occurring, all this development of this fibrous tissue, edematic tissue, and along with the inflammations, all these are occurring at such areas of the skin, and the such skin also started losing the sweat glands, and the skin affected area disintegrates the sweat glands, and all the total skin becomes rough. So as a result of that, that entire the region becomes enlarged, and due to the development of this fibrous tissue, and the skin becomes dry due to the loss of sweat glands, and due to the accumulation of the lymph in the surrounding tissues, and it also leads to the lymphedema. So lymphedema, development of fibrous tissue, loss of sweat glands, and the skin entirely becomes rough, and that enlarged parts, and whichever the parts in which the parasite actually gets infects. So that condition we finally refer to as elephantiasis. So finally it actually attacks at the extreme body condition like the limbs or scrotal sacs or the breast. These are the more prone areas and where the severe infections actually occurs. So all these together we call it as elephantiasis. So that is actually the entire the total life history of these parasites. And so we actually studied the life histories of the 4 different parasites which are responsible for the causing of disease. One is the Entamoeba histolytica, responsible for causing amoebiasis, Plasmodium life history, responsible for causing the malaria, and Ascariasis caused by the Ascaris lumbricoides. Finally the Filariasis or the elephantiasis caused by Wuchereria bancrofti. So these are all some of the protozoan parasites and the helminth parasites that we studied so far. And now let's move on to the some of the diseases which are also caused by the bacteria and also the virus and also by the fungus. And so some of the diseases that are kept for our study are, the one is the typhoid fever, which is actually caused by a bacterium called Salmonella typhi, which mainly lives inside the small intestine of man. So the presence of this bacteria can be get confirmed to the blood test, which is known as Widal test. So with which we can able to identify the presence of that parasite or the presence of the bacterium. And so how it actually occurs, and so as through the contaminated food and water only this bacterium enters inside the small intestine. So once it enters inside a high fever and very severe fever, almost the fever ranges to the 104 degrees Fahrenheit, and the stomach pain, constipation, the improper digestion, of course rise in the temperature anyway leads to the improper digestion, headache, loss of the appetite, and one of the more severe conditions is intestinal perforations, and the bacteria are responsible for the formation of the perforations inside the wall of the intestine, that sometimes it may leads to the death. And so every time the rise of the fever, we cannot able to suspect the it's a typhoid fever only. See, even in malaria also you will get the rise in the body temperature, and even in the Ucraria light infection also you will get the high fever. In typhoid also we will get the high fever. There are viral fevers are also leads to the fevers, but through the test called Vidal test, the Vidal test, the typhoid can be get confirmed. And then comes to the second type of the bacterial disease is the pneumonia, which is caused by Streptococcus pneumonia or Haemophilus influenza. And it is a gram-positive bacterium which is actually get infected mainly to the alveoli of the lungs, and so which is actually enters inside mainly by means of the inhalation of contaminated droplets, and which are coming out from the infected person, or else the sharing of the utensils, or the sharing of the kerchiefs, or whatever, any kind of the things that are used by that infected person by the normal person is actually there is a more chances of getting infected to this one. So it is responsible for the causing of severe respiratory problems, and of course and the breathing problems, and the lips and fingers, also nails, and they get turned into grey or the bluish. And these are the symptoms, getting of the respiratory problems and the breathing, along with the turning of the nails and into the grey or bluish. So one is the typhoid, and another one is the pneumonia, which we commonly get affected on. Move on to the another diseases which are caused by the viruses, and which are the viral diseases are, and these among these viral diseases, common cold, which is caused by the group called rhinovirus, and the rhinovirus group mainly infects the nose and also the respiratory passes, and but do not attack the lungs. And so many of the people actually misconstrue that this actually the common cold affects the lungs, and it nowhere get related to the lungs, it only the respiratory passes, and from the nostrils to the till the trachea or the bronchi only it is present. And now how it is get affects is, it's mainly spread through the contaminated or direct inhalation of droplets from the infected person, and through the also by means of contaminated objects. And now some of the symptoms are the nasal congestion, and difficult in the breathing, and the continuous discharge from the nose, and the throat is also get transformed into the harshness, and the symptoms almost last for the 3 to 7 days. And so as you know that if the cold, a common cold, whether you treat it or whether you don't treat for this one, it will get remain for the 3 days. So it's a common saying, and that is always present. Cold is treated, it can be cured in 1 week, or the cold is untreated, it will also remain for the 7 days. And it's like a small saying that used to say with respect to this common cold. And now another one is the let's move on to the fungal diseases, and it's a one disease that is given related to the fungus is ringworm, most common infectious disease caused by the these 3 different kinds of the fungus, and one is microsporum, another one is the trichophyton, and another one is the epidermophyton. These are the 3 different kinds of the fungi which are responsible for the causing of this ringworm. And if you look at the symptom that is appear with respect to the ringworm, see this as a development of the patches, and like the skin dry, scaly skin, whatever, there's different various parts of them, and the folds of the skin, groins, and also in between the hips, and these are the areas and where it's more prone to get this kind of disease. And so especially mainly get spread by means of contaminated towels, cloths, or the combs, and the infected person, even from sometimes from the soil, and the heat and moisture help and fungi to growth in case of the skin folds wherever you get these regions. And so dry scales and round lesions and small lesions are also formed accompanied by the Intensive itching on especially the regions of the skin and the scalp region and the nails, and these are all the regions which are prone to get the itching sensations, and the dry scaly skins with along with the round lesions are also formed. So these are some of the symptoms that are more commonly appear related to the ringworm. So these are the some of the bacterial as well as the viral and the fungal diseases, and that are mainly kept for this our study with respect to this under the topic health and diseases. And under the bacterial study diseases, we studied the one is a pneumonia and another one is a typhoid. Under the viral we have got the common cold, and under the fungal we studied about the ringworm. Under the protozoans we studied the entamoeba and the plasmodium. Under the helminths we studied the ascaris and ochraria life histories. Let's move on to the another important topic under the health and diseases, that is the cancer. Cancer is also one of the dreadful disease which is responsible for the causing of the death of several number of the people all over the across the world. And this is a condition which is in which the normal cells are actually keep on get dividing, but whenever the cells are get affected with this cancer, these cells are loses their cell division and become erratic and keep on dividing uncontrollably. And so this uncontrollable cell division we call it as cancer. So this is result in the formation of a mass of a tissue, or the mass of the cells that are formed as a result of the uncontrollable cell division are repeated number of cell cycles. So usually in normal cells the cell cycle regulation is present, and that's the number of cells that are should be formed is under check run. But whereas in case of the cancer, they loses their control on cell cycle and keep on dividing and proliferating and increasing in their number, resulting in the formation of a mass of tissue, that what we call it as a neoplasm. Or the abnormal proliferation of these cells results in the formation of a neoplasm, or which we call it as neoplasia. So these neoplasms are again get divided into the mainly into the two different types, and are the tumors we divided into the two different types. One is benign and the second one is malignant. Sometimes we can also classify into three different types like benign, pre-malignant and malignant also we can define it. So a benign, it's an harmful and it's not that much harmful, and they remain in the confined areas where the whichever the area this it is get affected, in that area only they are developed. So but as a result of this loss of the cell cycle and the several number of the cell cycles undergo and result in the formation of a mass of tissue, that what we call it as a tumor. But only the thing is that is confined to the one particular region, or the region where it actually get originated. So, so it do not get spread to the other parts of the body. So they actually cause a little damage to that part, the part where it actually get arises. For example, uterine fibroids, birds, they also forms as an best example for this benign cancers. So there the another classification, that is a pre-malignant cancers are also present, in which the cells started getting into the transforming into the cancerous. And so it's like a condition that before it is actually get transforming into cancerous, but it is not completely get transformed into a cancerous tissue, or in due course of the time it is going to get transformed into a cancerous condition. That condition we call it as a pre-malignant. And the second type is actually the malignant tumors. So malignant tumors, these are the one as a result of the loss of the cell cycles, they get divided and keep on dividing and result in the formation of a large mass of the tissue is formed, and which is called as neoplastic, and they keep on growing and rapidly, and also started surrounding the normal cells which are present around it. And one of the main important feature of these cells is they have provided with an important property, that what we call it as metastasis. Metastasis is a condition in which these cells started detaching from these tumors and getting spreading to the other different parts of the body. And that is one of the main important property of this disorder, that is malignant tumors. And not only that, these cells started competing with the normal cells, especially for the nutrients, and also started disrupting the metabolism of normal cells that are present inside the body. So And in these cells, of course, the cell cycle is lost, cell differentiation is lost, and the growth is also get lost. All these properties are actually lost in these cells. And one of the another important feature of these cancer cells is they do not exhibit contact inhibition and show the uncontrollable division. So actually, whenever the cells are keep on get dividing and trying to replace the lost cells inside the body and come in contact with the neighboring cells, the normal cells usually stop the division. That what we call it as contact inhibition. But these cancer cells do not show the contact inhibition property, and they keep on get dividing and developing into the large mass or the tissue. And later stage they started detaching from the site of the origin and spreading to the different parts of the body and giving rise to the secondary cancers. That what we call it as metastasis. So that condition is also actually seen. So once they get detached, they get started moving to the distant parts of the body, and where they get fixed and may give rise to the secondary cancers at the different parts of the body. That is one of the important feature of this malignant cancer. So malignant cancers are actually lethal, and whereas benign tumors can be simply get removed or eliminated from the body by the surgeries, and they are not that much severe or not harmful. But whereas malignant tumors, they are actually called as cancerous cells and leads to the death, and they are actually also lethal. And cancer cells also lack the anchorage dependence. And so when these cancer cells, usually whenever the normal cells are get dividing and developing as a tissue, all the cells are responsible for attaching with each other with the help of their cellular junctions. And as you already studied in the epithelial tissues, like they are provided with the desmosomes or the gap junctions, or with the help of the tight junctions. They all are each cell get binds with each other. But whereas in case of the cancer cells, they do not have, they lost their cellular junctions, especially the cadherin, the calcium dependent cell membrane proteins which are present, and they are actually lost, so that the one cell keep on dividing. These cells do not get attached with each other, so that they get exhibit the condition called as metastasis. So they get detached from each other and do not come to lie as a mass of a tissue. Then another thing is, another property of these cancer cells is the cancer cells actually lost the property called apoptosis. That is a self death or a programmed self death, we call it as apoptosis. This apoptosis phenomenon is also evaded by these cancers cells. And usually whenever any cells that are present inside the body that get transforms into an altered self cells like the cancer cells, they are actually get detected by these NK cells and destroy them. That is one of the important property of the NK cells, is to detect this altered self cells and destroy them. But these cancer cells are so cancer cells are also can able to escape from this NK cells and remain unrecognizable, and that is also the one of the property. And the another property of these cancer cells is these cells can able to increase the blood circulation and make them a new blood vessels route is actually get created, and make the nutrient supply towards them. That is what we call it as angiogenesis. So angiogenesis phenomenon is also get initiated by these cancer cells inside the body to draw the nourishment towards these cells, and decreasing the nutrient supply to the surrounding normal cells. That is also another property. And that is the reason why, because they are responsible for the stealing of the nutrients from the normal cells and diverting those blood vessels and the supply towards these cancer cells, these normal cells usually starve and die. So if you could able to stop this formation of the new blood vessels towards these tumors, so we can easily make the cancer cells to get die. Then another one is, what are these reasons and that are mainly responsible for the causing of the cancers? And see, inside the cells of the every cells of the living organisms are usually get provided with the genes that are responsible for causing cancers, and those genes we call it as proto-oncogenes or oncogenes. And interestingly, contrast to this one, we are also provided with tumor suppressor genes are present. But the proto-oncogenes, which are normal cellular genes that are present inside the body, and these genes are present in recessive condition. So, proto-oncogenes are present in the recessive condition only, and these genes get transformed into dominant as a result of a mutation. So, at least out of the two genes and these proto-oncogenes, if they could get transformed into a dominant genes as a result of the mutation, that is responsible for cause of cancer. But whereas to suppress these proto-oncogenes, we have got tumor suppressor genes are present, which are actually present in a dominant condition inside the genome. So these tumor suppressor genes are responsible for the suppressing the proto-oncogenes, which get transformed into oncogenes. So proto-oncogenes on mutation, they get transformed into oncogenes, and if these oncogenes are responsible for causing these cancers, and they get suppressed by tumor suppressor genes. And so as long as tumor suppressor genes are present in dominant condition, proto-oncogenes remain as proto-oncogenes without undergoing mutation and remain as proto-oncogenes only. So cancers, there is no question to get erasal of cancer inside the body. And so failure of the tumor suppressor genes or the undergoing the mutation of the tumor suppressor genes to get transformed into a recessive condition, proto-oncogenes from recessive to transform into dominant as a result of mutation to transform into oncogenes, then only we can able to develop, then only there is a chances of development of cancers inside the body. So another kind of the genes that are present inside our body are the p53 genes, and these p53 genes, it is a very much important genes that are present, and they play the important role also by acting as a G1 checkpoint, especially in the regulation of cell cycle, in cell cycle regulation, and p53. So that's why this p53 gene actually called as guardian angel of cells genome, because it guards the integrity of a DNA, and whenever the DNA undergoes the mutation or whenever it gets damages, so this p53 genes stops the cell cycle at that before the crossing of the G1 checkpoint, and make this gives a chance for the cell to get repair the DNA and again to continue the cell cycle. So like that it's play the important role. In addition to that p53 gene, and recently there is another gene is also get discovered, that is a retinoblastoma protein that is present inside the body. And so these are all the tumor suppressor genes that are present inside our body to guard these oncogenes, and if at all the proto-oncogenes get transformed into oncogenes. Then what are the agents or the substances that are responsible for causing of cancers? And so all those which are responsible for causing cancers are called as carcinogens, so which are responsible for damage the DNA. So there are the several number of radioactive substances are present, and which are responsible for causing cancers, and which can be a physical or chemical or the biological agents which bring about the uncontrollable proliferation of cells inside the body. So these carcinogens are of following types: the physical carcinogens, UV rays, X-rays, gamma rays; chemical carcinogens, aniline dyes, chemicals present in the tobacco smoke. And tobacco smoke actually consists of the benzopyrene or such type of the substances are present, and which chromium, nickel, arsenic, polonium. There are the various kinds of the heavy metals are also present inside the tobacco. All are responsible for causing of cancers. So sometimes apart from that, tumor viruses are the cancer causing viruses, which are called as oncogenes viruses, and which have got viral oncogenes are present, which are called V-onc. So C-onc and the V-onc. C-onc are the cellular oncogenes which get transformed from the proto-oncogenes. And now V-onc are the viral oncogenes responsible for causing of cancers. Now Another type is that there are the different types of the cancers are actually get classified based on their origin actually, and so they are the so for example the cancers which are actually cancerous condition that actually get developed in within the epithelial cells, they are actually called as carcinomas, and they are the most common type of cancers. And the second type of the cancers are sarcomas, which are actually developed in the connective tissues, and which are in case of the muscles also. If they are get developed within the blood or the bone marrow, then they are actually called as leukemias. And there the fourth type of the cancers are the lymphomas, and which are actually developed in case of lymphoid tissues or the bone marrow tissues. And again, cancers are divided into two different types, which are the familial and the sporadic. And if the cancers are actually responsible for passing from the one generation to the another generation, they are familial, and if they are confined to the one particular generation, they are not spreading, which are non-hereditary cancers, and so they are which do not have got any family histories, and they are called as sporadic cancers. And then coming to the how these cancers can be detected or diagnosed. And cancers can be detected by biopsy and histopathological studies of the tissue, blood and the bone marrow tests for the increased cell of leukemia, and the techniques like the radiography, MRI, CT scans, especially for the internal organs, and use of the antibiotics against the cancer, specific antigens for the certain types of cancers. And applying the principles of molecular biology to detect the genes individuals with inherited susceptible to the certain, especially for the certain types of cancers only, where we can able to detect those cancers. And so once the detection is get done, how the treatment can be given, and there the different types are there. For the combinations of all these are also responsible. For example, going for the surgery, tumor cells are surgically removed to reduce the load of the cancer cells inside the body, and the second one is the radiotherapy, and they are irradiated wholly, but taking care of the surrounding cells. The radiation is sent inside the body to especially kill the cancer cells. And then the chemotherapy, and especially certain types of the drugs are used to kill the cancer cells, and major drugs which have got a lot of side effects are present, like the loss of the hair from the body, skin, that what we call it as alopecia, and decrease in the number of the bloods, decrease in the blood number of blood cells, we call it as anemic conditions. Yeah, some of the secondary symptoms also appear in case of chemotherapy. And coming to the immunotherapy, this involves the use of biological response modifiers, like especially the alpha interferons, and which activate the immune system and also helps in destroying the tumors. And so nowadays we have got biological response modifiers like alpha interferons, which activate the whole immune system and make so that our immune cells can able to recognize these foreign cells or the altered self cells, and especially helping in destroying the tumors. And a recent advance in the science is a recognition of the cervical cancer, and the cancer that actually occurs or developed inside the neck of uterus, that what we call it as cervical cancer, which is actually seen in women in nowadays mostly, and which is actually caused by a virus called human papilloma virus. And this virus is for this virus because responsible for causing these cervical cancers, especially in women. So and vaccine is also get developed against for this one cancer. So as a prophylactic measures, a vaccine is also get developed for this for the eradication of this virus. And the women are actually get screened for the development of these cancers with the help of a small test called the pap smear test, in which a small tissue is get taken out from that cervix of the female reproductive system of uterus, and which can be get sent for the histopathological analysis, and the cancer can be get easily diagnosed. So in the diagnostic procedures, and not the like biopsies there or the histopathological studies there are the different methods are there for the diagnosis of these cancers. And even for the breast cancer, we have got mammogram is also there, with the help of that also especially the breast cancers can be easily get diagnosed. And whereas for the prostate gland cancers, which are also common in case of men, they also get diagnosed with the help of prostate antigen test analysis is also there. With the help of that also we can easily diagnose the presence of the cancers. And then the CT scan, MRI, they all are also diagnostic imaging techniques are available nowadays. With the help of that, they also can able to detect the cancers. And so especially the another type is the PET scan is also there, that also detects the cancers, and which also helps in the physicians to get diagnose the breast cancers, or which are get developed inside the colorectal cancers or the cervical cancers. All they can be easily get diagnosed. And there are another kind of the method is monoclonal antibodies techniques are also present, and biological drug morphites, they are also useful, especially in giving the treatment to the certain cancers, that what we call it as a biotherapy. And the biotherapy is also nowadays available with the help by especially treating those cancers with the help of monoclonal antibodies. So the detections and treatments, there all are the different types of the methods are there, especially to treat these cancers. And